Good afternoon, everybody, and thank you for joining us for today's UCD's Institute of Food and Health Research Bite. So today our talk is going to be on healthy oats, and I'm delighted to welcome Dr. Andrea Silva Espinoza. And Andrea is a postdoctoral researcher in the School of Agriculture and Food Science. She completed her PhD in Food Science and Technology in the Polytech University of Valencia in Spain. And while she was there, she also did part of her PhD as a visiting student in the University of Nottingham. Her PhD focused on the development of new plant-based products using fruits. She selected the correct formulation, the appropriate processing conditions to achieve the end good sensory properties and physico-chemical properties with the highest amount of bioactive compounds with good physicochemical attributes as well. As a result of her PhD research, she published 7 publications, so it was a very productive PhD for Andrea. As I say, now she's working on a project which is termed Healthy Oats, and Professor Fiona Doohan is the overall leader of this project, and Andrea is going to share some of the work that's been done in Healthy Oats, and particularly her own project. Over to you, Andrea.
Thank you very much, Dolores, for the introduction. I can presume all you can hear me well. So my name is Andrea Silva. I'm a postdoctoral researcher working here in UCB in Healthy Oats project, under the supervision of Professor Dolores O'Riordan. And today, my presentation is titled Healthy Oats, Advances that Support New Oats Product Development. So I'm just going to do a quick overview of my presentation today. First, I will do an introduction to Healthy Oats project. Then I will do an overview of our research program. Then I'll move on to background to research and progress to date. And finally, I will mention the future plan of our research. So let's start with Healthy Oats project then. So Healthy Oats project is a cross-border collaboration research project between Ireland and Wales that is funded by the European Regional Development Fund that promotes the development of oats as a healthy food product and a climate-resistant crop. So traditionally, oat is a crop ideally suited to the climate of Ireland and Wales and well established in the traditional farming systems. In addition to that, looking for those new oat varieties that are climate resistant also contributes in tackling different aspects and challenges like the environmental one, which is also linked to the more sustainable food production, like reducing the use of pesticides and also reducing the nutrient losses. Another factor to take into account in terms of, for the purpose of Healthy Oats projects, is that currently lifestyle population go towards healthier diets. It gives different opportunities to food industry to diversify their products, especially going to more plant-based products. In the latest years, oats have gained considerable notoriety due to some interesting properties that presents the oats with huge potential for new product development. And some of these oats properties is that it's the cereal with the highest protein content. It has beta-glucan. The beta-glucan is a soluble fiber that is related to health benefits, such as lowering the cholesterol, reducing the risk of heart disease, and also has gastrointestinal effects like increasing the satiety and has prebiotic activity. The oats also present polyphenol compounds that is related to the potential antioxidant activity, which has been reported to prevent some pathologies. Under this context, the aim of Healthy Oats project is to enhance the innovation and productivity in the Irish and Welsh food production sector through the development of high-quality oats for new and improved food products. So, Healthy Oats project covers what from farm to fork approach is called. And that's why Healthy Oats is a multidisciplinary project. The work packages working in Healthy Oats project covers different aspects like Work Package 2 on the germplasm, disease, climate resistant and nutritional evaluation covering the agronomical fields. Then Work Package 3 covering the consumer behavior and health studies, and Work Package 4, which covers new product development, which is where my research has been focused on. So I wanted to represent Healthy Oats project as a year system because the different work packages are in constant communication and collaboration to accomplish all the objectives and tasks proposed by Healthy Oats project. So yeah, so in terms of the research program, our objective is to create the know-how and understanding required to create that added value for potential new oat products, but also for the ones that are already in the market, for the health and well-being market. So to achieve this objective, we have been working in different tasks. So the first of them is to establish the processing conditions for the new oat varieties to ensure physicochemical properties that will maximize their functionality through the analysis of nutrients, stability, and microstructure. As a second task is to assess the technofunctional properties of the processed new oat varieties like the solubility, water retention capacity, rheological properties, and swelling volume, and also to assess the stability of the new oat varieties during the storage. And as a third task is to establish formulation and processing protocols to manufacture prototype food products containing new oat varieties like oat drinks, protein oat balls, and non-dairy products like cheese. So yeah, to accomplish these three tasks, our work comprises two areas. The first of them is the physicochemical analysis of oat products, and the second area is the oats processing. So first, I'm going to start talking about our work on the physicochemical analysis of oat products. So first, what we did is to set up standard methods to assess composition and physicochemical properties of oat products, taking as references official methods as the Analysis American Association of Cereal Chemists and the Association of Official Agricultural Chemists. So different methods were set up for biochemical composition, such as for macroelements, fat, protein, starch, moisture, ash, amylose, amylopectin ratio, beta-glucan, and phenolic compounds. And also methods were set up for physical properties related to technofunctional properties, such as size distribution, swelling volume test, solubility, and water retention capacity. So different samples were tested, including conventional and organic oat flakes, extensively for their physicochemical properties. Regarding the sampling, the samples were provided by Flagavants as a result of our first industry day organized by Healthy Oats projects, and also because Flagavants is strong in the Irish markets, and that's why we can use these samples as a benchmark samples to be compared with the new oat varieties. So in this table, we can observe the results of the characterization of the biochemical properties for the different samples for both conventional and organic oat flakes. So we can observe here the mean and the standard deviation for each of them. So in general, the results are in agreement with theoretical values published in literature, which confirm that we can use these samples as a benchmark samples. Going a bit more in detail, we can observe that the organic oat flakes showed a significantly lower amylose content and higher total phenolic compounds than the conventional oat flakes. This is relevant because, as I already mentioned, the total phenolic compounds have potential antioxidant activity, which has been reported to contribute in the prevention of some pathologies like cardiovascular diseases, some cancers, and some deterioration processes. Regarding the technofunctional properties, I would like to highlight this result, which is that the organic oat flakes showed significantly lower water retention capacity than the conventional oat flakes. Also, a positive and significant correlation between the amylose content and the water retention capacity was observed. So all of these results suggest that the oat growing conditions has an impact on the macrocomposition, polyphenols, and the technofunctional properties of the oats products. So yeah, now I would like to move on to the second area, that is the oat processing. So, and I would like to highlight the importance of the oat processing. So the oats are non-digestible seeds that need to be processed in order to be able and stable for human consumption. So here in this flow diagram, we can observe the different steps during the oat processing. So, which includes the cleaning and the grading, the dehulling, which is the step where the husks are removed. And then it comes the kilning, which is the steaming step where the vapor is injected to the oats, and then a drying step or the roasting step as well. So once we get the groats heated and stable for consumption, they can follow further steps like flaking, grinding, cutting to get different oat products like oat flakes, whole oat flour, oat bran, and steel cut oats. So also we have focused our work in the heating treatments because they are critical steps. And why are they important? Well, because they are applied to inactivate oat enzyme systems. It means the lipase, the lipoxygenase, peroxidase, since the oats are susceptible to the development of bitter oat flavors and rancidity due to their high fat content. Also because they develop the nutty brown toasted oat flavor and aroma characteristic of the oats, and also because during these steps, the conditions, it means the time temperature combination, affects the final physical quality of these oats and therefore their functionality. And one example of application of the oat processing that we are working on it is the oat drink. So there is increasing demand of plant-based beverage, in concrete of oat drink. However, based on practical and theoretical evidences, there has been reported some flavor issues in the oat drink that they might be due to the oat processing conditions. But how do we get the oat drink? So, well, let's say that we can divide the oat drink processing in two parts. So the first part is the one that includes the dehulling and the heating treatments, which is basically the part that all the oats need to go through independently of the final product that you want to get. So in this first part, there are two types of oat processing. The typical or older one, where the kilning, steaming step is carried out before the dehulling, and the more modern one, where the kilning step is carried out after the dehulling. So this difference in the oat processing could be affecting the final quality of the product of the oat drink, and therefore can be responsible of these flavor issues. So what we did is we compared both of them. So if we follow in this flow diagram, the orange line, we can observe that we have the typical oat processing where the kilning steaming is carried out before the dehulling. And if we follow the blue line, we have the modern oat processing where the dehulling is carried out before the kilning steaming. So also we increase the time of drying roasting from 20 to 100 minutes to see this how this factor have an impact. And then the objective was to get the growths with between 9 and 10% of moisture. After that, we did a characterization of the flavor profile, lipid oxidation, and lipase activity. So with this experiment, the aim was to evaluate the effect of the dehulling before and after kilning process, and also to evaluate the effect of the different processing conditions of the kiln growths on the flavor profile, lipid oxidation, and lipase activity. This can be directly applied to oat industry as an opportunity to improve oat processing to achieve high-quality oat-based products. So some of the key messages of our results is that the steaming conditions applied increase the moisture for oats with and without husks. And the difference for both samples of moisture was just 0.5%. Also, that the steaming conditions must ensure the activation of lipases in both of samples. That is important to avoid rancidity during the storage. Regarding the increasing heating time during the drying roasting process, as expected, the longer time of drying, the lower moisture of the oats. There was no benefits in heating beyond 20 minutes in terms of inhibiting lipid oxidation and lipase activity. Also, this allows to save energy and minimize the loss of nutrients and bioactive compounds like the total phenolic compounds. The desired final moisture content was obtained after just 20 minutes during the roasting process. So yeah, based on these results, we established the oat processing conditions for the new oat varieties, assuring the lipase deactivation, the reduction of lipid oxidation, and also the moisture content target for every step. Since the moisture content is relevant, since it's directly related to the enzyme deactivation during the process, and also it will affect the grinding process and therefore the oat functionality. So once we get the growths, they could be ready to be consumed. However, they usually go to additional steps to get different oat products. And in Ireland, one of the most popular and typical oat products is the oat flakes. So what we did is also to establish the conditions for the oat flaking processing. So, and the oat flaking includes 3 different steps: the steaming, the flaking, and the roasting or drying. So the steaming has the objective to increase that moisture to reduce the fragility of the oats and therefore avoid breakages during the flaking or rolling step. Then during the roasting or drying, the objective is to reduce again the moisture to make the oat flakes stable from the microbiological point of view. So let's say that this is the first part of the oat drink process. So the oat flakes actually can be, is a final product as we know it, but also it can be used as an intermediate product to prepare other products like the oat drink. So, and this is what we are doing now, we are working on it. So in this flow diagram, we can observe the oat drink processing, but this is just the process for exclusively for the oat drink. So this process includes the addition of water to the oat flakes, a hydrolysis step, the decantation, filtration, formulation with using different ingredients, homogenization, and heating treatments. However, to establish those processing conditions, we need to take into account a few parameters and attributes to get a high quality oat drink. And just to do an overview of those parameters, like for example, during the addition of water, the ratio oats-water will affect the texture and flavor. During the hydrolysis, that has the objective of avoid starch gelatinization in further steps, and also to offer that adequate sweetness to the oat drink. The type and the concentration of the enzymes and also the time temperature combination during the hydrolysis will affect to all these parameters. During the formulation, we can use these ingredients or some of them, like for example, the oil, sodium chloride, calcium carbonate, some vitamins, and some emulsifiers, that it will have an impact on the creaminess, texture, viscosity, flavor, and also to the final use of oat drink. You might have seen in the market that they usually have 2 versions of the oat drink. So they have a barista version and let's say the conventional, the normal version. So for example, the barista one, it has a higher concentration of oil because it needs that creaminess when to have like a adequate final product when it's mixed with other products like the coffee or the chocolate. During the homogenization that has the objective to make the emulsion stable, the pressure and the number of passes through the homogenizer will have an impact on the mouthfeel, the color, and the stability. And finally, the heating treatments, which has the objective to make it stable from the microbiological point of view. the time temperature combination will affect the shelf life of the oat drink and also the storage conditions. So I wanted to show you just some of the pictures that's of our work in the oat drink. So we can observe here, we are using the oat flakes. And then once they are mixed with the water, they are filtrated. We can observe here the oat slurry after the filtration. In this picture, we have the formulation pre-homogenization steps where the oat slurry is mixed with the different ingredients. And then we have here the high pressure homogenizer. And in this picture, we can observe, well, I don't know, I think you can observe, but this is the sample that is that's already passed through the homogenizer, through the high pressure homogenizer. And the right one, which is a bit whiter, is the one that is after the homogenization. Yeah, so it's clear, we can clearly see that the color is affected. So yeah, finally, I would like to mention some of the future plans of our research to evaluate the effects of processing of the new oat varieties and its impact on oat drink, to establish the storage stability of oats from various processing regimes, meet with industry, and also liaise with World Package 3, responsible of consumer insights. Finally, I would like to especially thank to Flahavans and Sons Limited, to Amalia and Puja from World Package 3, to Cajal from World Package 2, and to the whole Healthy Oats team, including the collaborating universities and institutes, the University College Dublin, Aberystwyth University, Swansea University, and Teagasc, Ireland. So this is our contact details in case you want to reach to us. And thank you for listening. I will be happy to answer any question if there is any. Thank you so much.